This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 426, recorded on January 27th, 2017. This episode of TWiV is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get your first 3 meals free with free shipping by going to blueapron.com/TWiV. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Welcome.
Thank you. It's a typical cloudy winter day, but it's 30 degrees Fahrenheit and -1 Celsius.
Yeah, we have a lot of clouds here, and now I have to use this new weather app because the other one broke, so it's kind of disorganized, but it is, where's the information? Where's it? Ah, there it is, 6 degrees Celsius. Can't see the sun, it's cloudy. It has been above freezing all week. Kind of a strange, we had a lot of rain on, what was it? Monday and Tuesday. It was just pouring. I guess nor'easter. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. I'm northeast of you.
Did you have a nor'easter?
We kind of did. It was rainy and slushy and just mixes of different things. And now it's blowing about 20 knots out of the west and some scudding overcast and low-level wind shear and just really turbulent weather.
Are we supposed to get some snow? Is that right?
I think so. The chance of snow for tonight.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
Good, very
All
good.
right.
So we got 56 Fahrenheit. That's 13 Celsius, and sort of a high overcast. Sort of a, meh day. I must say that this morning there were scattered high clouds, and the sunrise was spectacular with all the pink lighting them up from the bottom. It's great.
So hopefully everyone will recognize or sense the difference in Rich's voice. He has a new mic, and now everyone has new microphones and new headsets.
Yeah, I'm feeling like a real, this is like a new day in podcasting, a real mic in front of me. This is great.
So thanks to all of you out there who have supported us and allow us to sound a little bit better. We do have a winner of the Infections of Leisure book contest. I feel badly. This one came in a few days ago, but the emails keep coming in, and they won't stop until Sunday when the new episode's released. But that's the way it goes. Our winner is Matt, who writes, "Greetings, TWiV and TWiV. As an MD student and longtime listener, I'd love to be the lucky recipient of Infections of Leisure. Sounds like a great supplement to my clinical training." And Matt is a MD candidate in the class of 2017 at the University of Miami Miller School of Medicine.
Good luck in the match, Matt.
Yes.
Excellent.
And I'm happy to have medical students here listening to podcasts about viruses. It's great.
Absolutely.
I know that your time is precious, but it's great. So congratulations. We're going to have another contest. So stay tuned, everyone. I have a brand new book right here next to me to give away. There's also a contest over on TWiM. We're not at the. It was just released yesterday, so we're early on. If you want to try your luck at that, Kathy Spindler, what is happening with the American Society for Virology?
The annual meeting will be June 24th to 28th in Madison, Wisconsin. This is the last announcement that you will hear about submitting your abstract because the deadline is this coming Wednesday, February 1st. That's also the deadline for travel award applications. If you want to apply for a travel award, you need to be a member. So you also need to be processing your membership, your travel award, and your abstract. I'm hoping that you've been listening for a while and you haven't procrastinated and you don't have all of those things to be doing on Wednesday afternoon. We really have to be very strict about that deadline because the abstracts all go into FASEB and we can't We can't take any late abstracts. The meeting site is asv.org. Click on annual meeting and then annual meeting, or go to the TWiV page for the show, and then Vincent's been putting the link to directly to the annual meeting in the show notes. That's another way you can find it. I think that's all I want to say about it. Oh,
if you think you're going to get the result that's going to change your abstract between now and Wednesday, you're wrong. So go ahead and do it.
Do it.
Yes.
Absolutely right. All right. I want to tell you about the American Society for Microbiology. They are putting in place a new governance to the people who take care of the society. And one of the most important steps is filling at-large positions. They're going to have a board of directors and a council on microbial sciences. And ASM members can get involved in leadership positions. So the board of directors is the government's body that sets the course for the society. It fulfills all the fiduciary roles, like approval of the budget. They are responsible for the society. So it's kind of like a board of directors of a company. And, if someone decides to sue ASM, they're suing the BOD. Wow. Council on Microbial Sciences, on the other hand, is the main advisory body to the BOD, and they'll make recommendations for the strategic, scientific, and programmatic direction of the society. Both bodies will serve key roles in the microbial sciences by providing leadership and direction to the society and to the whole field. There's going to be one slot on the BOD and one slot on the comms for a young microbial scientist, someone at the level of grad student, postdoc, assistant professor, or junior clinical microbiologist or equivalent. There'll also be a slot in the BOD and two on comms for members working outside of the U.S. It's going to be a nicely representative two different bodies. It's really good. So I'm going to put links to a number of things in the show notes. First, those of you who are ASM members, you can vote for these individuals. I'll put a link for the voting. And then some informational stuff. What positions are we voting for? What is the board of directors? What is the Council on Microbial Sciences? And some organizational charts. So if you're an ASM member, you have the right to vote every year. You should exercise it and do that. It's really important. It determines the future of your society. We have a follow-up from Bridget. Hello, Vincent and the TWiV crew. Thanks for picking two articles about the value of publicly funded science and the hard work climate scientists are doing to preserve their data. Sadly, in the last week, things have started to look even rougher for some government scientists and science funding. Some people in our field have looked at the successful women's marches that happened on January 21st, and they're planning something similar for supporters of science. And she provides a link to the basic event outline, though most of the real planning activity is taking place on social media. Links to Facebook, Reddit, etc. are on that page. I hope you encourage more listeners to keep standing up for science. While the main march will happen in Washington, D.C., there will likely be sister marches throughout the U.S., so listeners throughout the country should keep an eye out and an ear open. Thanks. I hope everyone's spring 2017 semester is off to a good start. So this is, on Twitter, it's Science March DC, well, one word. Yep. And depending on which Saturday it is, they haven't decided yet, I'll be there. Okay? I usually can go on a Saturday. There's no podcasting or anything else. My kids are all in college, so my wife's a scientist, so maybe we'll both go. But I'll be there. I hope to see you all there.
What spring 2017? Can we narrow it down any more than that?
They haven't announced the date yet. They just set this up a few days ago. And it, I think, probably took off a whole lot faster than they expected. Kathy put this update in that they had 244,000 followers as of 2:05 today.
Mm-hmm.
Wow!
And they now have 246,000. I checked it about half an hour ago, and they had 245,000. So they are growing very, very rapidly in interest. It's gotten some news coverage. And they have a message at the top of their Twitter stream saying they're going to announce the date in the next few days.
That could be interesting.
Now,
yes.
I spoke to someone here in New York City, and they told me they are overwhelmed by the response. So I hope they can get it together because these things are not trivial. To have a lot of people going to a city is not trivial.
Yes, that was my first concern when I saw this was, who are these people and do they have any clue what they're doing here? And it seems like they're doing kind of the right things. They're putting up Google Forms for people to sign up for stuff, and they they actually seem to be drawing in the expertise or possessing the expertise that they need to get this thing put together.
Yep. Well, we'll see, because the Women's March in D.C., highly organized. You need permits, right? So they had, let's see, 1,200 buses. They parked in the parking lot at the football stadium, and they had everyone take the subway in or walk. It's not that far. Right. And then you have to have walking permits and all that stuff. So really, really well organized. And so this has to be as well. Otherwise, it turns into a disaster and doesn't look good for science, right?
Right.
But you have to have a good sound system if you're going to have speakers.
Yep.
That was a concern I had for the Women's March in Ann Arbor, but they did have a good sound system, so that worked out.
Yeah, I am hopeful about this, but yes, the folks organizing it need to stay on top of it.
It's not.
There's a website for this too. I just found.
Yep.
Yes.
So you can
scientistmarchonwashington.com.
Yeah, it's Bridget sent the link to that in her email.
This was actually going to be my pick this week, but I saw that it was picked.
I just, down in the Twitter stream, I saw a cartoon of somebody holding a sign saying, "Science is real. Your alternative facts are square root of -1."
Yeah, well. All right, we have a snippet in the paper today. The snippet has to do with prions again. We have talked about prions a couple weeks ago, and this paper has just been published in M-Sphere, the open access publication of ASM, and I thought it would be worth talking about briefly. The title is De Novo Generation of a Unique Cervid Prion Strain Using Protein Misfolding Cyclic Amplification. And I think listeners will remember we talked about protein misfolding cyclic amplification a couple of weeks ago as a way to detect prion proteins. And so I thought this would be good to talk about it because it's fresh in your minds.
So is this where you're the I editor?
This is where I'm the I editor.
So you are now executing your function as I editor.
I'm executing it. That's
okay.
Right. I get a weekly list of the accepted papers, and I look at them for podcast interest. Yeah. So this one I picked, and there's another one coming out. I think we're doing one on TWiV, or we did one on TWiV. I can't remember. Yeah. So now and then I pick one. And this is by, the first author is Crystal Meyer-Reed, and the last author is Mark Zabel. They're from the Prion Research Center at Colorado State University, and also the College of Veterinary Medicine there in Fort Collins. So basically what we have here, many of you may remember, but prions are proteins, and they can cause disease when they misfold. So diseases like Creutzfeldt-Jakob is a chronic degenerative neurological disease where your prion protein, you have a normal copy of the prion protein, it misfolds spontaneously, it propagates, it causes other copies of the normal protein to misfold, and when you get enough of those accumulating in your brain, you develop the neurological disease. And variant Creutzfeldt-Jakob, which we talked about a couple of weeks ago, occurs when you eat meat, beef contaminated with prions from mad cow disease, bovine spongiform encephalopathy.
Or you get it into your bloodstream because you're butchering meat.
Butchering meat, or get it somehow. Now here, the topic of this paper is a cervid prion. Cervids are like deer,
elk,
moose, and elk, which lovely name, cervids.
Yes.
Like it. But
reindeer.
Reindeer too.
Yes.
Reindeer too. And they call this chronic wasting disease. It's a prion disease of cervids. It affects these cervids that we just mentioned. And it's a relatively recently discovered disease. It's endemic in both free-ranging and captive herds. Okay, so deer walking around in the woods, and also anything you got in your in the back of your farm there. North America, South Korea, and very recently Northern Europe. And where it came from is the question. So some people think, the deer or the elk or the moose could have been infected by sheep. So there are some sheep in these areas where, except for Europe, there have been sheep in North America, South Korea. And people think, well, maybe sheep, they get scrapey and they somehow transfer their prions to the deer.
And that was based partly on the correlation between a TSE, a scrapie outbreak in sheep, and the initial discovery of chronic wasting disease in Colorado and Wyoming, which was 1960-something, 62 or 63. So it was a little while ago.
I should also remind everyone that I think maybe we did a paper a long time ago. If not, I blogged about it, that plants can take up and transmit infectious prions. So you have a sheep walking around in a meadow. It's excreting prions. They can be taken up by plants. Then if a cervid comes along, it could eat it and could be transmitted that way. And of course,
the species barriers on these has always baffled me, because scrapie, from my understanding, is not readily transmissible to humans.
Right.
But is transmissible to cattle, correct?
Yeah, that's right.
Yeah.
And in cattle, you get BSE, and that is transmissible to humans.
That's right.
So with the intermediate species, now you have something that can infect humans. And of course, in all cases, you come in with a prion from one organism that is or is not capable of templating itself on the native protein from the other organism. So I would presume that what happens in the new species is what you get is a little different than what came in. All right. And so that accounts for the
that's right.
transmissibility through another species. But it's bizarre, the routes.
So in fact, if you take a cervid that's died of chronic wasting disease, you make an extract of the brain, you inject it into mice, they don't get disease. But if you make the mice transgenic for cervid prion gene, PRNP, then they will get disease. Cows can be infected with cervid prions, right? You don't have to make
so would those be deer mice?
So you don't have to worry about making cows transgenic, but of course, cows are out in pastures, and you can imagine that maybe overnight deer have been there defecating or urinating, and there's prions in them. But cows eat them, they get contaminated, then someone eats the cow, and they get sick. So this is a problem for the food chain. So this paper tries to address where cervid prions came from. What's the origin of chronic wasting disease? As I said, it could be sheep, but in Europe, the recent outbreak or the recent cases, there are no sheep that are evidently involved. So could it happen spontaneously? In people, you get spontaneous Creutzfeldt-Jakob, where you're just walking around and all of a sudden your prions misfold and catalyzes more misfolding and you get sick. So they do the protein misfolding cyclic amplification, which you may remember, you take a little bit of the native protein, the prion protein, you mix it with a misfolded prion protein that you get from an infected animal. And the misfolded protein will cause misfolding of the seed, and you can do multiple cycles of folding and sonication to break up the oligomers and then more folding. And it basically amplifies a small amount of misfolded protein, and that's why you can use it as a diagnostic case test. But here they ask, can we get misfolded cervid prions spontaneously? So they do PMCA, and the measure for a misfolded prion is a Western blot. These when the protein misfolds, it becomes resistant to protease digestion. You can run a Western blot, and you can see that readily. And so
by spontaneously here, what we mean is they're doing these amplification incubations without seeding it with any prion protein.
With any pathogenic protein.
Right, so they're just taking healthy protein and cycling it through this process. It's in vitro, but it's
and reading the paper, it's reasonably clear that one of the things that inspired this is that, of course, a cycling reaction without seeding any prion protein would be, under normal circumstances, a control experiment for other PCMAs. And it's clear that they got occasional false positives out of their controls. And this is one of those moments that's funny.
Right.
Yeah. It's not good because, for a blood test, you don't want to have false positives, right? And even
so, are there really false positives?
Yeah.
Or is something going on?
Well, here they just do, as Rich said, PMCA with wild type protein, which they can produce, as recombinant protein, and they get after 4 rounds of this amplification, they got 1 protease resistant prion protein, which then they would increase in subsequent rounds of amplification. So it can apparently spontaneously misfold in vitro, and you can detect it in the PMCA. And these misfolded prions are infectious. They take transgenic mice and they infect them with these proteinase-resistant prions generated in vitro by this amplification method, and the transgenic mice get sick. So here we have 4 out of 5 mice died. It took 273 days. Wow. You have to be patient.
That's fairly typical of prion experiments.
You have to be very patient. Yeah.
Yes.
And that's compared to 8 out of 8 mice that die when you inject them with an extract of deer brain who died of chronic wasting disease. 285 days, 8 out of 8 mice. Okay, so you can make misfolded deer prions in vitro. That's the bottom line, really. And these are distinct from the prions you might get from a deer who naturally died. They have different glycosylation profiles. They have different conformational stabilities. They even cause different patterns of neurological disease in the CNS of mice. So not only
you could say, well, that's just because the in vitro system is not equipped with the same glycosylation enzymes, et cetera, as the in vivo. Or you could say it's because the in vitro system is just not a good representation of the in vivo. See?
Yeah. Now remember, these proteins are all the same sequence.
Yes.
So they're conformational differences and glycosylation differences as well. And that's really the bottom line, that you can get, at least in vitro, you can spontaneously misfold this cervid prion. So presumably in nature, it could also misfold in a deer. And maybe that's how it originated once, a long time ago, misfolded in one deer, and then it got propagated to other deer and maybe to other animals. So it could just arise in different parts of the world spontaneously.
And it could arise repeatedly, which is important for explaining some of the patterns that have been seen in the wild, where you'll see this in a particular area. And by the way, it's called chronic wasting disease because the deer starve to death, basically, and they have neurological problems and they don't feed properly. But they come wandering out of the woods and dying in significant numbers, and then after a while, this seems to go away. The number of affected deer declines, and then things seem okay, and then it breaks out again sometime later. So that could either be waves of infection coming into the area, or it could be new spontaneous development of this in another deer.
And they make the point that based on the structure of this thing and on their experience with the in vitro results and on the experience in nature, as Alan described, that this particular prion may be more prone to spontaneous transformation to the pathogenic form of prion than some other prions.
Right.
And so it's interesting that cervids are the only free-ranging wildlife affected by TSCs that we know of anyway. So nothing else. I mean, I think everything out there is free-ranging, right? Rodents and all that, they're all free-ranging, right?
Right. It may simply be that in order for us to notice something like this, it has to be a species. It has to meet a bunch of requirements. It has to be a species that we care about, that we would notice dying, and that we would see the way in which it dies. I mean, something small like a mouse or even a rabbit or a squirrel. If the rabbit in the backyard starves to death, I'm not going to notice. It's
oh, you're so mean.
Well, I think they're cute, but they'll wander off and disappear, and maybe a hawk got it. And it also has to be a species that lives long enough.
Yeah. So I wonder, there are no reports of of non-human primates, right? Getting prion, like chimps or gorillas. Maybe some of the death in nature is due to this, but we just don't know.
It's got to be. These diseases have got to be in all these animals.
Yeah. It might be hard to detect, though, yeah.
Mm-hmm. Yeah.
Well, so that's our snippet. I thought that was a good follow-up. Make a little arc out of prions.
Yes.
Okay. Anything else before we move on? Is that good?
Yeah.
Good.
I think that's a real... We actually snippeted that.
Yeah, it's very rare to do a snippet.
We managed.
Yeah. Over on TWiV, we have problems keeping it to snippet length.
Yeah, we need a shot clock or something.
This episode of TWiV is brought to you by Blue Apron. Blue Apron's mission is to make incredible home cooking accessible to everybody while supporting a more sustainable food system and setting the highest standards for ingredients and building a community of home chefs. They deliver seasonal recipes with fresh, high-quality ingredients so you can make delicious home-cooked meals. Each meal comes with step-by-step, easy-to-follow recipe card. It's one page, nice glossy card with nice pictures on it. Pre-proportioned ingredients. The whole thing takes less than an hour, 40 minutes or less. If you spend a lot eating out or at high-end grocery chains, you can now spend under $10 per person and make healthy home-cooked meals. They set really high standards for community. They have a community of over 150 farms, fisheries, and ranchers. These are local establishments, so you get local deliveries. For example, seafood is sustainably sourced under standards developed with the Monterey Bay Aquarium Seafood Watch. Beef, chicken, and pork come from responsibly raised animals. Produce is sourced from farms that practice regenerative farming. And they ship you the exact amount you need for a recipe, reducing food waste. And often they'll have spices that you don't have, so you don't have to go out and buy a whole bunch. It's fresh. Get everything except salt, pepper, and oil, basically. It's really nice. We've done a few of these now in the last couple of weeks, and they're just easy, fun, and tasty. Very good to do. And you can customize your recipes every week. Choose a delivery option that meets your needs. There are meals for 2 people or 4 people, for a family, for example. There's no weekly commitment. You get them when you want, and they deliver to 99% of the continental U.S. You can choose from different recipes every week, and they don't repeat any recipe in a given year, so you don't get bored. For example, recent recipes have been spicy shrimp and Korean rice cakes with cabbage and furikake, pork chops and garlic piccata with scallion rice and spinach, cashew chicken stir fry with tango, mandarins, and jasmine rice. So you get a meat or a fish, you get a vegetable and a carb. Check out this week's menu. Get your first 3 meals free with free shipping. Go to blueapron.com/TWiV. That's blueapron.com/TWiV. You'll love how good it feels and tastes to create incredible home-cooked meals with Blue Apron, so don't wait. Blue Apron, a better way to cook. I think for all of you who are really busy out there and love to eat but don't have a lot of time to cook, this is really a good thing, so check it out. We thank them for their support.
I just put in an order, and I'm supposed to get the box on Monday, so...
You'll like it. You'll like it.
I really think so.
Good. I mean, you end up making these gorgeous dishes that are just very tasty. It's not hard. All right.
And they have plenty of dishes that are vegetarian as well.
Mm-hmm.
So it seems like all the ones that you have in the list to read are non-vegetarian, but
yeah,
you can get all 3 of the meals to be vegetarian.
Oh yeah.
For instance, there's one I had that was potato and artichoke quiches. Just lots of good options.
Yep.
Yeah. And you can choose. I think I picked 2 meals for the first week that I'm doing this, but you can choose like 2 or 3 or 2 or 4 or something.
2 or 4, yeah.
Yeah.
I think you get 2 for 2 people. You get 3 meals, and for 4 people, you get 2.
Okay.
In a given week, something like that. Yeah. All right. We have a paper for you published in Cell Reports, which is the open access publication of Cell Press. It's entitled "Axel Mediates Zika Virus Entry in Human Glial Cells and Modulates Innate Immune Responses." This is a topic which is interesting. We've talked a bit about Axel as a potential receptor or entry protein for Zika virus, and I think this has got some interesting data in it. And we haven't talked about Zika in a while, so it's still out there, and people are working on it. This is a paper. The first author is Laurent Meertens, and the last author is Ali Amara. It comes from a variety of places in France and French Polynesia. For example, the Hôpital Saint-Louis which is in Paris, the Université Paris Diderot, the Pasteur Institute, the Institut Louis Mallardet, which is in French Polynesia, Université Paris Est, and the CNRS in Montpellier. And finally, in Houston, Texas, the Ruga Corporation. All these wonderful places, and then Houston, Texas.
Houston, Texas is a wonderful place.
Is it?
Yeah, there are people who live there.
Well, the people may be good.
I think we'll get letters.
But I don't know. I was there once, and it seemed pretty polluted. I don't know. And hot and humid, but that's not bad. I agree. Houston, Paris, I don't know. Given the choice, where would you go? Anyway, Zika virus, everyone...
French Polynesia, that's where I'd go.
Everyone remembers that Zika virus emerged, the end of 2015. During 2015, it became a global issue and infected large parts of Brazil and was associated with microcephaly and other neurological issues in fetuses, a little bit of sexual transmission, and a lot of people had started to work on it. Largely, I think, because of the microcephaly connection, because otherwise it was previously a rather benign rash-like joint pain type illness. So we have many, many labs working on it, trying to understand the basic biology and pathogenesis. So here's one where it's been previously suggested that AXL, a protein on the surface of the cell, could be involved in entry of Zika virus into cells. And there have been a variety of papers already published on this, beginning with papers where they overproduced AXL in cells, and that was correlated with increased infection by Zika. A couple of papers have shown that antibodies to AXL or knocking down the mRNA with small interfering RNAs would reduce virus infection, and also the presence of AXL in cells that are targets of virus infection in humans. All these were some evidence that AXL was involved. Now, we discussed, let me tell you a little bit about AXL. AXL is a member of a family called the TAM family. Tyro3, AXL, and MER are three tyrosine kinase receptors in this family. They're involved in the clearance of apoptotic cells and regulation of innate immunity. And it's been previously published that dengue virus binds to AXL indirectly through a protein called GAS6, which is a ligand of AXL. The way this happens is that GAS6 recognizes phosphatidylserine on the viral membrane and bridges the particle to the AXL receptor. And then the virus, so the virus dengue virus at least binds GAS6, which then binds AXL, and then it gets taken up into cells. So in this publication, they're looking in human embryonic neural tissues and some cell lines to see what's going on with AXL. There's
a little bit of a controversy around AXL as a Zika receptor. There
is a bit of a controversy. Yeah.
So the flip side of this is that there are some studies showing that you can knock out AXL and still infect with Zika virus.
Yeah, so we discussed on TWiV 406 a paper from Cell Reports also where they had mice lacking AXL gene. It was knocked out. And
right.
These, if you infect them with Zika, the virus will still replicate in the eye and in the brain. Right. So in some situations, as you'll see today, AXL seems to be involved in entry and others not. So there's probably more complicated than simply one. Also in organoids, human organoids, they can remove AXL by CRISPR and the virus will still replicate. So this is an in vitro kind of cell culture system. So yes, and then in other studies, it looks like it might be involved in entry.
And it may be something like the virus will use AXL if it's there, but if AXL is not there, there's an alternative pathway that it can use.
Perhaps, yeah.
So just a little more about the normal function of AXL, because I was a little confused about that, and I found this nice. They refer to one of these mini reviews in Host Cell and Microbe by a guy named Jason Mercer, and that has a nice cartoon in it with a nice legend that shows the normal function of AXL and how that's co-opted by membraned viruses. And AXL is thought to be involved in clearance of apoptotic cells. And when cells apoptose, they butt off bits of cells that have phosphatidylserine on the surface. And as Vincent said, this GAS6 can bind the phosphatidylserine and the AXL and trigger the uptake of those cell bits for digestion. And using the same mechanism, Gas6 can recognize the phosphatidylserine on the surface of a virus and take it up. So ordinarily, it's something that would presumably help clear apoptotic debris.
Right. Now,
and so that's a question I had, is so that these viruses just by chance have some phosphatidylserine in their membrane, and wouldn't that often be triggering things that maybe wouldn't be advantageous to the virus? Did I miss something?
I think it's a common constituent of the cell membrane, right?
Yeah.
So if you're budding from the cell, you're going to get phosphatidylserine.
Yeah.
I thought it was the inside-out thing or something. I don't know. Okay.
Yeah, well, phosphatidylserine, I think, is usually my understanding. We're going to get mail here, but that's good. Is that it's quite often on the inner leaflet of the membrane, and part of the dying process is that it gets externalized, and that's a signal for eat me. Now, Jason Mercer, the guy I mentioned before, is a pox virologist. Okay, so that's how he got into this, because he illuminated this pathway for pox viruses. And interestingly, the pox virus membrane, which is a fairly novel membrane, is rich in phosphatidylserine on the exterior. And it's been shown that if you specifically strip off that PS from the viral membrane, it loses its infectivity. And you can actually reconstitute the infectivity by feeding phosphatidylserine back to it. So it's clear that the phosphatidylserine on the outer portion of the membrane on that virus is important for this.
So because it's on the inner leaflet in normal cells, the presence in virus particles you would assume is there because of the cells have undergone apoptosis and it's flipped?
Or the budding process itself may have the effect. Now, I'm making this up, but the budding, and actually I'm cheating because I'm looking at Jason's cartoon here, which implies with a lentivirus, for example, that the budding process itself might be involved in placing the phosphatidylserine on the exterior of the membrane.
So the enzymes involved, they're called flippase and scramblase. That exchange of phosphatidylserine. So on cells, the presence of phosphatidylserine triggers engulfment by macrophages. And so I wonder if that is a viral evolutionary thing to get it taken up into cells.
That would make sense.
Right. Now, in this paper, what they do is they get 20-week human tissues from abortions or dead embryos and cut the brain parts up and look for AXL production by immunofluorescence.
This is happening in France, remember?
Yes. It's happening in France. They have permission to do this. It's in the methods. And they find at 20 weeks strong axle in microglia, which are mainly in the ventricular and subventricular zones. So if you have to think now, when the brain is developing, in the center there's the canal, and around it is the ventricular zone. And there are lots of radial glial progenitor cells, which are like stem cells for neurons. And they're eventually going to become neurons and then move up glial tracts to the pia, the outer surface of the brain. But there are also lots of astrocytes and microglia in this area. And they find axle in the microglia, the radial glial progenitor cells. And did I say microglia? Yes, astrocytes, microglia. So,
and do I understand correctly that astrocytes and microglia are sort of the maintenance staff of the brain?
Yeah.
Okay.
They're the brain's immune system.
Okay.
Well,
they also do stuff like
they do other stuff,
myelination and stuff, right?
Yes.
Yeah.
Astrocytes are really important for the blood-brain barrier, and I think they're also important in neuron development, whereas microglia are the resident macrophages.
Okay.
So I don't especially think of astrocytes as being immune cells functions. Endothelial cells in the brain have some immune cell function, but I don't associate that with astroglia per se.
And these cells aren't specifically associated with the developing brain, are they? They're in adult brains as well.
Yes.
Right.
Oh, right.
Yes, they are. All right. So they ask, can these cells be infected with Zika? So they have a microglial cell line. These are all human cells so far. Human microglial cell line. They have primary human and marine astrocytes. And they have human neural progenitor cells, which you can make in vitro from stem cells. and differentiate them into neural progenitors, and they infect them, and they measure infection by flow cytometry throughout this paper. So they have an antibody to a viral protein, and you can tell if the cells are infected. And I'm a little unhappy with that because they haven't a single assay for infectious virus. You would think at some point they would say, okay, this flow signal corresponds with infectivity, but it's never done. And I don't like that. I think you have to at some point say there's infectious virus here.
Right. Otherwise, you're just following fluorescent labels around.
Yeah. I mean, who knows what's going on? So they used an African strain. This is a derivative of, I think, the first Ugandan strain. They infect these various cell types. And what they find is that the microglial cell line, the human and murine astrocytes, and the neuroprogenitor cells can be infected. Then they say, how about an antibody to AXL? What does it do? Infection is blocked by this antibody in the human and murine astrocytes and the microglial cell line. And also silencing AXL does the same thing. But antibody to AXL doesn't affect infection of human neuroprogenitor cells. So right away you see there's a dichotomy here. There's some other receptor probably, because AXL is not blocking infection of these human neuroprogenitor cells. So I think that's the important message that we're getting from all this work. There's probably more than one way for Zika to get into a cell, depending on the cell.
More than one way to skin a virus.
Didn't we use that as a title once? I think we did.
Maybe.
I think we did. Now, they also show that dengue virus can infect these cells, the microglial cell line, murine astrocytes, and the neuroprogenitor cells in an AXL-dependent manner, which has been suggested before as well. Okay, so next they ask what role the ligands for AXL are playing in Zika virus infection. There are two known ligands, Gas6 and protein S. And they do a very simple infection where they can grow these microglial cells in the absence of fetal cath serum. And apparently that has these ligands in it. So if you grow cells without, there's no TAM ligands, there's no AXL ligand. And then in those conditions, Zika infection is reduced. And if you add back Gas6, you restore normal levels of Zika virus infection. So, this suggests that Gas6 is binding to the virus and also binding to AXL. Although I don't know if anyone was bothered by just leaving out serum, if there wasn't something else in there that could also be participating. I don't know. Wouldn't you like to see them pull out gas specifically instead of just adding it back?
I would be happier with that, but adding it back really kind of helps. I mean,
I know there are subsequent experiments that further reinforce this, so I guess it's okay, but I just would like to have seen that. They have a decoy called MYD1. It's a decoy receptor that has high affinity for Gas6 and blocks its interaction with AXL. So the idea is you've got Gas6 and AXL. Now you add this decoy, and it should basically sop up Gas6. And when you do that, it prevents Zika infection in a dose-dependent manner. No effect on herpes simplex virus, which doesn't require AXL for infection of cells. And this works in the microglial cells, in HeLa cells that produce AXL, and in primary astrocytes. So looks like the virus is binding gas, which is binding AXL. Although I would have liked to seen a direct gas virus binding, which they didn't do. But everything points to that. Everything points to that. Antibody to Gas6 also inhibits Zika virus infection. Okay, so that now the idea is Zika, gas, AXL. That's the order of interaction. They next looked at knocking down AXL expression in these microglial cells. They use CRISPR to do that. And as you might suspect, in AXL knockout cells, they don't get Zika virus infection.
Which is in contrast to work in other cell types that's found
exactly
knocking that out prevents Zika virus infection. So
yes,
different models.
And they block when you remove AXL seems to be internalization because you don't get as much RNA internalized in these knockout cells. They also want to know how the virus is brought in via AXL into cells, and they ask, well, is it clathrin-dependent endocytosis, which is a kind of endocytosis pathway, so they can knock down clathrin, and it would inhibit that pathway, and that strongly blocks Zika virus infection. So virus is binding Gas6, then AXL. It's taken into the cell by clathrin-mediated endocytosis. And the last thing they ask is, are these early or late endosomes that are involved in release of the virus into the cytosol? And there are two markers that they use for this. There's RAB5 and RAB7. Which one is which? RAB5 is an early endosome. RAB7 is a late endosome marker. They knock down each of those, and they find that it's early endosomes out of which the virus is exiting in this path.
So something that kind of confused me here. They have a sentence here that says AXL is known to mediate phagocytosis of apoptotic cells, yet flavivirus entry occurs mainly through clathrin-dependent endocytosis. So does that mean that, phagocytosis and clathrin-dependent endocytosis are different pathways, right? Mm-hmm. After the original binding.
That's right.
So does that mean that in this particular case, the virus is somehow legislating trafficking through a pathway that's not the normal pathway for AXL?
So the phagocytosis makes sense because when phosphatidylserine is exposed on a cell surface, then the macrophages see that, and then they take up the cell by phagocytosis. Right. So it's presumably AXL on the macrophage that's doing that, right?
Okay, so maybe it's cell-specific
I think
that's going to happen.
That could be.
Okay.
Yeah. All right, so AXL is a protein kinase, a tyrosine kinase, as we mentioned earlier. They want to know if the protein kinase activity has any role. So you can make a single amino acid change within the AXL protein, so it's catalytically inactive. They ask, what does Zika virus do in cells with such a protein? Well, the virus can bind. It can get into cells that have catalytically inactive AXL, but the virus doesn't replicate. So you need this kinase activity, which, by the way, happens when gas binds to AXL. If you have a wild-type AXL, you add gas, you get activation of the kinase activity. I think Rich mentioned that earlier. And the same thing happens when you add Zika and gas to AXL, you get even more phosphorylation of AXL. And they have a small molecule inhibitor of the kinase called R428. R stands for Rich, no doubt.
Yeah,
and that inhibits the kinase activity and also inhibits Zika virus infection, but doesn't block entry. So there's something going on after entry that the kinase activity is involved in.
And this may suggest a resolution to this this conflict in the literature of AXL's the receptor, AXL's not the receptor. It seems like AXL is doing a couple of different things, and maybe in some cell types you can complement some of those with other proteins, and in other cell types you can't.
It could be, but in any case, in the absence of AXL, if you still get infection, it's got to be through some other entry pathway, right?
Yes, there's got to be another entry factor, but we now see there could also be this kinase activity, which, maybe AXL can do that, or something else can do that.
Yep.
In some other cell.
Now, the last set of experiments, very interesting. It's known for some time that AXL, when AXL is activated, the phosphorylation starts a signaling cascade which dampens the innate immune response. It suppresses the innate immune response. It does so by inducing a protein called SOX1, suppressor of cytokine signaling 1, and this in turn suppresses interferon responses. And of course, interferons would be needed to modulate virus infection. So they looked at interferon and SOX1 in cells infected by Zika, and with or without this AXL antagonist R428. So again, that's the kinase inhibitor. In the presence of kinase inhibitor, this leads to a increase in both interferon and SOX or mRNA levels, and also some other interferons as well as pro-inflammatory cytokines, other interleukins. So basically, when the virus hits AXL, it may suppress innate immunity by turning on SOX, which in turn suppresses the interferon response. All right, so that's brilliant. The virus not only can bind to Gas6, which binds to AXL and gets it into the cell, but in turn it activates the kinase of AXL, which in turn shuts off or turns down innate immunity, and that would let the virus replicate better.
Right.
I don't know any example of that where, but it just may be my general ignorance, but I don't know an example of where a virus binding a receptor will also down-modulate innate responses. Does anyone know?
Not chapter and verse.
Chapter and verse.
It does sound. It has a familiar ring to it, but I don't know of any specific examples. I have a little hallucination here because I'm wondering what AXL is doing, dampening the innate immune response anyway. Mm-hmm. And I'm thinking, so triggering the innate immune response is sort of that's a big project for cells that causes distress down the line, even death. Mm-hmm. And I'm thinking that here's a receptor that at least part of its function is to import garbage and get rid of it. And that garbage could have all sorts of immune modulatory stuff in it that could have lousy consequences down the road. So maybe the idea is, okay, we're going to take on some garbage here. Let's turn off that inflammatory response while we deal with this. Mm-hmm. And then we can turn it back on.
Yeah, I mean, in general, when you turn things on, you got to turn it off again at some point. Mm-hmm. That's what the SOX proteins are involved with, right? Suppressing all this. Because if you don't, it's going to be on all the time or a long time, and that's bad. So
right. You may want to turn it off while you're eating junk.
Right.
Exactly. Well, that's basically the paper. And remember, so in some cells, the first part is that it's an interesting way for a virus to attach via a Gas6 that then attaches to to AXL, brought into the cells by endocytosis. So that's that. The second part is that the binding activates AXL, which seems to suppress innate immune responses. And the third is that blocking AXL doesn't prevent infection of neural progenitor cells. So there's probably another way of the virus getting into cells. So, in the end, this will all be explained and clear. But until then, people say, oh, it's this in this paper and that in the other paper. But there's always an explanation, I guess.
Quite often, people get into their thing and they want to say, this is it. People are always looking for a unique solution. And quite often the answer is all of the above, Life
is a lot messier than we want it to be.
Yeah. And the other point.
But you're right. People get really invested in their own thing, mm-hmm. and they don't want to listen to other viewpoints. And that's a whole part of science, is to listen to what other people are saying and say, oh, yeah, this all makes sense if you look at it this way.
This is why we have and need science, because the natural human reflex is to want to win the argument. And
we have a president now who wants to win all arguments.
Yeah, I mean that can be productive if it's channeled in certain ways, but the key to science is that you want to be correct. Yeah. You don't want to just win an argument, and you want to be correct so that you can win an argument. But ultimately, it's about getting at the truth, and the way we do that is by hashing all this stuff out and saying, look, it does this over here, it does this over here. Let's work out, what's going on and why. Mm-hmm.
The other point they make in this paper is that they've got a couple of inhibitors of this process. One is the MyD1, which is a little cumbersome because that's a big receptor in itself, but the other, R428, is a small molecule inhibitor. And so that raises the question of whether you could use anti-AXL or anti-Gas6 agents such as R428 to inhibit a Zika infection. At the same time, they make the point that if you do that, you're tinkering with some pretty lively molecules, and it may have bad side effects. So, at any rate.
I mean, I think here the biology is what we learn about, right?
Yeah.
Yes. This is not directed towards therapeutic intervention. I don't know why every paper they have to say this could be a potential target, but
because they want to get money.
It's terrible.
Yes.
That's not the only reason, as you know. But I know the study sections have made us do this. What's the human application? Come on, it's knowledge.
Well, and it's too bad that the study sections, because the study sections are scientists as well. They need to take a look at themselves and say, "Wait a minute, what are we doing here?"
Yeah, and even the way that last sentence of the summary is written, it's really that inhibiting axil function represents a potential approach for future antiviral therapies. It's not representing a potential target for future.
Yeah.
Anyway, just
yeah.
But
I just want to complain very briefly. I find these papers very hard to go through. The figure legends are really minimal. You have to look all over to figure out how they're done, and so much of the data are in the supplementary data. I just dislike this very much. I think it's a really poor reading experience.
I agree.
And I don't know why they have to do this. Why can't they make it easier for people to read this? Can you imagine an undergraduate trying to read this paper and figure out what's going to take them days?
Yeah.
Well, because Cell wants you to read their flagship journals, and they're not sure that open access is going to be a good business model for them, so they're putting minimal effort into it, is my theory.
It's our science, man. I don't want people doing minimal effort with our science. Think about that, folks. Hey, I use the word folks.
Yes, good job.
It works.
It does work. Folks is pretty good. All right, we have email from Stefan, who writes, Dear Vincent, Dixon, Kathy, Alan, and Rich, my name is Stefan, and I'm a junior doctor from Edinburgh, Scotland. I'm a longtime loyal listener to the Twix podcast. I first tuned into TWiV back in 2010 when I was an undergraduate studying infection and immunology at the University of Oxford, and I've been hooked ever since. The chance to win a copy of Infections of Leisure is the carrot that finally prompted me to write in. I hope I'm lucky number 17. And he has a listener pick, which I think we will move to later. Someone remind me if I forget. Thanks for the podcast, and please keep the weather updates. It's nice to be reminded that although Scotland might be cold, it rarely reaches the sub-zero temperatures of southeast Michigan. P.S. I'm enjoying my PDF copy of Parasitic Diseases 6th edition, so thanks also to Dixon and Daniel for making it free to download. All right. Kathy, can you take the next one?
Becky writes, Hi, chaps. I hope I'm not too late for the book contest. Even if I am, I just wanted to say thank you to you all for the great work on TWiV. I'm a biomedical scientist by trade, starting in a hospital diagnostic virology lab, and thanks to everything I learned from your podcasts, I got the confidence to go for my virology Master of Science degree. I'm now doing a part-time PhD alongside a full-time job at Public Health England, working on hepatitis E virus, which you really should do an episode on. I also volunteer for the London Virology Discussion Group, and I recommend your podcasts and lectures to all that attend our evening talks. By the way, if you're ever in London, Vincent, we'd love to have you. We'd love to host you for an evening. I've gotten so much from your kind efforts, and I'd love to give something back. I heard on the last episode, 424, that you are wanting to transcribe your podcasts to make them searchable. I'm happy to transcribe some episodes for you by way of thanks for all the great work that you make freely available. If you let me know which ones you have transcripts for, I'll get to work. Many thanks, Becky. P.S. I almost forgot the weather. A cloudless sunny day here in Colindale, London. Temperature 2 degrees Celsius. So my bet is that we don't have transcripts for most podcasts.
Yeah, there is a page somewhere with transcripts on them, which I will post in the show notes. I haven't looked at it for a while.
Big job.
It is a big job.
Big job.
Yeah, it is a big job. Alan.
Paul writes, Hello, Twivers. Hopefully I'm the 17th email. I'm always looking for a new book to read. The weather in Chapel Hill, North Carolina is currently a pleasantly sunny 59 Fahrenheit, 15 C. A little background on myself. I'm currently an epidemiology PhD student at the University of North Carolina, hoping to specialize in infectious disease epidemiology. I've always been excited about virology. I've been listening to the podcast since episode 402 after a fellow student and former student of Vincent told me about the podcast. As for a listener pick, I would like to mention the book The Panic Virus, The True Story Behind the Vaccine Autism Controversy by Seth Mnookin. I've recently read it, and I thought it was especially relevant in light of episode 423's discussion regarding speaking with lay audiences regarding vaccinations and RFK Jr.'s meeting with the president-elect. best Paul. And Paul, actually, Vincent, what episode was that you had Seth on?
117.
117, thank you. Oh, yes, and Kathy's noted it down here.
We had him on the show, right?
Yep, we talked to him for a couple hours.
Yeah, you should go back and
check out 117. Good episode.
Yep.
Actually, when I introduce people to TWiV, that's one of the episodes I recommend because you don't have to be. It's very low tech, and he was a great guest.
Rich.
Oh, me. Jamie writes, "Hello, all. Ever since I heard you mention the Infections of Leisure book, I have been hooked on getting it somehow and hope I might be the lucky 17th and win it. Sounds like a great read that would help keep my micro knowledge relative as I take a hiatus. Relevant, okay. As I take a hiatus from my career as a clinical microbiologist to be a stay-at-home mom. Even if I don't win it, I am especially thankful to you guys and gal for bringing science into my daily life and keeping me in the know. Today is a cool, damp 47 degrees F here in Vernon, New Jersey." Good.
As you can see, the book giveaways get a bunch of emails, which is great. We like to hear from different people. Johnny writes, "From my hometown. Who knows? Wouldn't a virus look beautiful as a mirror ball?" So she sends a link to an article. Yolanda Baker, the last of the disco ball makers.
It's an amazing site. There's video there, and just watching her make these things on a spinning thing, and some of the disco balls are giant, and there used to be many people making them in this factory, and now she's the last one, and she's going to retire.
Oh, there's a niche. There's an opening.
There you go.
Right.
And she has
Where's she hang out? This is in Boston?
Must be in Boston, yeah.
Can I move to Boston and make disco balls? I'll do that.
Sure.
Well, you could have another house there since you have kids there, right?
Yeah, right. I'll just go live with my daughter, make disco balls in her basement.
Yeah, she'd love that. Johnny also sends a YouTube video, Sky Spiral or Leaving the Lollipops. It's a kinetic wind sculpture by Anthony Howe.
These are really cool.
Very neat. They're very good stuff, yeah. And Johnny says, suggest viral structures to me and hints at the fourth dimension. Yeah, these are very cool. I got to turn my sound off here. Lovely. I'd like to have one in my backyard.
By the way, this is totally aside, but there was an episode called More Than One Way to Skin a Virus. It was episode 175.
There we go.
And we had Matt on.
You're next, Kathy.
Megan writes, "Hello, TWiV crew. I'm writing from Redlands, California, where it's currently 15 degrees Celsius, 59 Fahrenheit, and partly cloudy. The link below is from the BMBL 5th edition and describes containment recommendations for working with prions in the laboratory. It confirms what you said in the podcast." And she has an excerpt from the text on page 284. "In the laboratory setting, prions from human tissue and human prions propagated in animals should be manipulated at BSL-2. BSE prions can likewise be manipulated at BSL-2. Due to the high probability that BSE prions have been transmitted to humans, certain circumstances may require the use of BSL-3 facilities and practices. All other animal prions are manipulated at BSL-2. However, when a prion from one species is inoculated into another, the resultant infected animal should be treated according to the guidelines applying to the source of the inoculum." And then they have a number for you to call APHIS National Center for specific guidance. And she says, "Thank you for all the work you put into the weekly podcast. I'm a virologist by training and studied 3 immune evasion proteins found in guinea pig cytomegalovirus for my dissertation. I have been listening to TWiV ever since episode 1 during a short postdoc. My career changed paths after that, and I'm now a laboratory director in a county public health lab. I love listening each week to get a peek at what's going on in the research world." And she's in Riverside, California. Oh, and she says she's writing from Redlands, but her job is in Riverside. One thing that came up when we were talking about the biosafety levels, just because something doesn't have a treatment or a vaccine or a cure doesn't necessarily elevate its recommended biosafety level. So, for example, there are a number of BSL-2 agents for which there's no treatment or vaccine or cure, but they're still BSL-2. So that kind of explains this BMBL recommendation for most of BSL2 work. But then, those are always only recommendations, and your local biosafety committee can require higher or can even allow lower recommendations if they so choose. That the latter would be pretty rare, I think.
Yeah.
But particularly based on the kinds of things that you're going to be doing. So, for instance, when we were talking about the sonication, then, yeah, that might require something higher, or a local biosafety committee might recommend something higher.
Yeah. So here,
and also the something else that's covered, I think, in the CDC publications is the sterilization procedure for things that might be contaminated with prions, which actually is kind of scary. Apparently, to inactivate these things, the preferred procedure is to autoclave them in bleach.
Right.
Which will apparently it'll destroy prions with fair reliability. It'll also destroy just about absolutely everything else in your laboratory.
Right.
So it's not clear that any non-disposable equipment should ever be used in these sorts of things.
Yeah, prions to me are pretty creepy. And the notion, the notion of playing with these at BSL2, especially the ones that can affect humans, is kind of gives me the creeps. On the other hand,
I'd be careful. Yeah,
BSL2 practiced as it is described is pretty safe. I think most people most people practice a pretty lax BSL2. Okay, but BSL2 is actually a fairly high containment level. And BSL3 really institutes practices and procedures that make it much more difficult to do stuff.
Yep.
Right.
Alan, can you take Nathan's, please?
Sure. Nathan writes, Dear TWiV esteemed hosts, I hope I'm letter number 17. Thanks so much for the opportunity to participate in TWiV 421. The TWiV studio should be a stop for science tourism in New York City. Well, I think it's becoming that. It was great to have the TWiV experience in real time and in vivo with Vincent and Dixon. Attached is a fascinating paper from Nature about communication between phage. Erez et al. 2017, and he gives the link. Query, does it reopen the question of are viruses alive? That question is always open here on TWiV. I suggest for either a snippet or a full discussion. Then following another recent story arc, here are a few of my favorite science podcasts. From the Twix family, TWiM and TWiVo, CDC's Emerging Infectious Diseases, Nina Martin's Public Health United, Cell Podcast, the eLife Podcast, the Nature Podcast, Genentech's Two Scientists Walk Into a Bar, and Gimlet's Science Versus. Weather in Chapel Hill, North Carolina is 36 Fahrenheit, 6C, and sunny. Keep on podcasting. And apparently, Vincent, you've decided you're going to do the communication paper on TWiM.
Yeah, we're doing it next week, so it'll be out in a couple of weeks.
This looks like a very cool paper, and I'm jealous you're doing it on TWiM.
Yes, me too.
Can you do it twice? Can you do it here too?
Yeah, if you want. It's I thought about it, and we could have easily done it here. It's not a paper that requires microbial expertise, but I just thought viruses of bacteria could go there. It's bottom line is it's a phage of Pseudomonas. When it infects cell, it encodes, the phage genome encodes a protein that's processed to a 6 amino acid peptide that's secreted extracellularly. When this peptide is brought into a new bacterium, it binds a cytosolic receptor, which then changes the response of the virus from lytic to lysogenic. So without the peptide, the infection is lytic. When the peptide accumulates, it becomes lysogenic, and the idea is that the level of the peptide is a marker for previous infection. So maybe if the population of hosts is getting low as a result of lytic infection, this thing will, say, go into lysogeny.
Right. So it's kind of a quorum sensor for phage.
Yeah, sort of. Although, this is a bacillus. Is it a bacillus? Yeah, it's a bacillus phage. The soil is full of bacilli. I don't know that you're ever going to have a shortage of them. So I'm not sure when this, this is another idea of another situation where you're applying human values to what phages do, and I'm not sure it works out. Maybe there's some other reason for it, not just population control. Anyway, it's an interesting paper. Do it next week. Rich.
Justin writes, hello, sages of TWiV. I've been listening for over a year now and greatly enjoy listening to the TWiX podcasts on my commute, as well as when performing the menial parts of bench work. The TWiX podcasts help keep me well informed and always give me good papers to discuss with my lab mates, as well as give me ideas of what I may want to pursue after my PhD. My fiance is an ecologist and has recently finished a book called Foundations of Ecology, Classic Papers with Commentaries. It gives a link. The book contains 40 classic papers in ecology that set the stage for modern ecology with commentaries. As a micromolecular biology PhD student, I have been incredibly jealous of this book and have been unable to find something similar for my field. I figured if it existed, the TWiV crew would know about it. So how about it? Is there a compilation of classic papers in virology, microbiology, molecular biology? Any of the above would be amazing and would certainly deserve my money. The weather here in upstate New York has been another unseasonably mild winter so far. This morning, it's an overcast 39F with a high of 48F, 4 and 9 degrees C, respectively. Best and many thanks for the hours of entertainment. Justin, who is a PhD student at the University of Albany. So my familiarity with Kathy's got some links here, but I know of no book like this. When I was a graduate student, they had a book that was classic papers in biochemistry or molecular biology or something like that, which I thought was fantastic. I have thought that somebody like me ought to actually put this together, but of course I've done nothing. Kathy, what do you got?
Is Kathy gone?
Oh, man.
Been awfully
we
quiet.
lost her.
Sorry. I remember a book that maybe the one that Rich was remembering also, and I remember it as having actual images of the original papers.
Yes.
In the book, but
yes.
I can't dredge up.
You mean a virology one or something else?
No, it was more molecular biology.
Yeah, it was molecular biology or biochemistry, and it was classic papers. It had the Watson and Crick paper. It had a bunch of others in it, but this would be a gas to do for virology. Actually, Justin, keep poking us because it would be fun to do. You know, Jenner, Pasteur,
right?
Ivanovsky, Reed, all of these guys, all of these papers, Rouse. There's just wonderful papers that could be done that would be absolutely fascinating.
All right, so I have 2 books here from my bookshelf. Now, the thing here is that nowadays all you have to do is do a compilation of links, all right? You don't have to reproduce the entire article anymore unless it's not open access, you know, and then it gets hard because you got to get everyone's permission. Anyway, I have 2. One is called Papers in Biochemical Genetics by Jeff Zubey and Julius Marmer.
I think that's the one we're thinking of.
Oh, and this is full of classic Okazaki, Kornberg.
Yes.
You know, John Cairns, isolation of an E. coli strain with a mutation affecting DNA polymerase.
Yeah,
that's full of cool stuff. And then I have a second one, which is very good also. Readings in Mammalian Cell Culture by Robert Pollack. It would sound like it's not all about cells, you know, it's about transformation, growth of cells. So we got here a method for titration of HeLa cells, the serial cultivation of human diploid cell strains by Hayflick and Moorhead. And
the Hayflick paper is a classic.
You know,
great
chromosome structure, mutant cell lines, enucleated cells, heterokaryons. So this is a good one. This is by Robert Pollack. So those are the two I have.
You should put those both in the show notes because people could at least check them out at their library. It sounds like the kind of thing that would be out of print at this point.
Maybe I should
did those have commentaries in them or just the papers?
They do. Some of them have commentaries. Yeah.
Does that biochemical genetics paper have an orange cover?
It's like psychedelic. It's blue and red.
Okay.
It's a second edition. Jeff Zubey used to be here at Columbia. Let me see if there are commentaries here. You know, there's usually there's a little preface to the section where they talk about things.
That's the way it ought to be done. The paper, and you ought to have a little preface that tells a little bit about the author, a little bit about, this is really getting me worked up here, a little bit about the author, a little bit about what the situation was at the time. what they knew and what they didn't know, including, what was available to them technically, because this kind of stuff is fascinating. Great.
So that's the key, that what's known and what wasn't known at the time that's going to require some work, right? Mm-hmm. It's not especially if you aren't familiar with it. Some of us can think back and remember, but really, really fun work, because it's an exercise in putting yourself in their shoes. Mm-hmm.
So the other thing that I found was I just Googled for classics in molecular biology and classics in virology, or classic papers in, and I got links to PNAS classics, Cell 40 foundation papers, landmark papers, Nature, the top 100 papers, and then from Elsevier, the virology most cited articles, which isn't necessarily classics, but so there are other ways to find those kinds of things as well. But it's these
so I'm starting a new document here that will never go anywhere. That's going to start with your links, Kathy.
Okay.
As inspiration.
I mean, you could have themed collections too. That would be useful, right?
Yes.
I'm really interested in sort of the ancient history. Well, not all ancient. I mean, reverse transcriptase. That's one of them.
Yeah, but I remember you talking about the Virology Journal Club at Florida, where you were doing the really old papers.
Yeah, we did a bunch of classics, and we did a couple of classics in the Stone Age here on TWiV. We did Jenner, and I think we did Pasteur. I think we did the rabies paper.
Could still do that. We could pick some old.
Oh, I love that. I just love that stuff.
Sure. Who read that email, by the way? I forgot.
I think I
did. You read it?
Yeah.
Yes.
All right, so I'm next. Michael writes, I've been running behind the past couple of weeks and thought surely it would have been over by now, but as soon as I started this week's episode, I had to pause and write this. Even if I don't win, I most sincerely thank you and the whole crew as always, and if I do win, I thank you that much more. You're running behind what, Michael?
The past couple of weeks.
Oh. Kathy.
Chris writes, hello, Vincent and the TWiV gang. I was introduced to your podcast last year while finishing my undergrad degree at Indiana University Southeast. The professor who taught my microbiology courses referenced the Microbe World site and your podcast several times, and I've been hooked ever since. I am currently working through the application process for acceptance into a physician assistant program, and listening each week has been a great way to keep my mind focused on many different topics and research in the field of virology. I haven't been able to listen regularly for a few months as our first child was born right after Thanksgiving, when I quickly learned that infants aren't fans of their parents' podcast time. I did, however, catch this week's episode and heard you mention that you were giving away a copy of Infections of Leisure, which I would certainly enjoy reading. I'm not sure what the requirements are to enter beyond sending the 17th email, but if that is the criteria, I thought I'd give it a try. Keep up the good work on TWiV, and I'll keep trying to adapt to tracking the Ohio Valley weather in degrees Celsius, which is no small task given how often things change here. Thanks, Chris.
Alan, you're next.
Sure. Livy writes, hello, TWiVerity. Your podcast is pleasurably infectious. Stumbled upon it like I do with most of my own virions accidentally while performing my own personal research on the microbes that affect us. I also watch Vincento's YouTube virology class. I like that, Vincento. In 2015, my immunity was suppressed by the love of a beautiful person, lowering my defenses to the infiltration of HSV1, acyclovir, and HPV. In a lecture, you mentioned virions are obligate molecular parasites. Every solution must reveal something about the host as well as the virus. Much has been revealed to me. About me, you don't have to read this, but we will. Kenyan student in America, Oklahoma, adventuring and experiencing this nation. Hope someday our African continent invests as much in science research and education to enable us to cope with nature's challenges. Well, pretty soon Africa may be investing as much in science and education as the United States, but not because, well, anyway. It's, hey, Alan, I'm a flight student too, and do it when I can, but you know the Oklahoma wind is seldom patient. De Pomieri, another good one. One day I hope to fill the whole sub-Saharan landscape with urban gardens. Gone are the days of African starvation, thanks to you. Kathy, lovely voice. You make me want to enroll in immunology and join your field. I like that you call the researchers and find about their lives and paths to science. Rich sounds like a great guy to hang out with. Planning a trip to Florida at the end of the month. If it works out, a run-in would be in order.
Well, yeah. Actually, at the end of the month, I might be in Florida.
Okay.
There's a regatta in St. Augustine. I'll be busy.
That's at the end of February? Or at the end of
March.
January?
Sorry. End of March.
Okay. But he's not that far from you if he's in Oklahoma. I mean, given
there you go.
It's an adjacent state to Texas.
Relative to Texas, yeah.
Right.
I like, he tells us how to pronounce his name.
Yes.
He says, good luck with my last name.
Wirigi?
I don't know if he would say the M at all. Wirigi?
Rigi?
Oh, well.
I'm trying to think about, somebody, a doctor I know, and I can't even think of his name, but it has two consonants like that, and can't remember whether you say them or not.
Rich, you are next.
Okay.
It's Scott's.
Scott writes, Dear TWiV, I wrote to you a few months ago to thank Vincent for the Virology 101 series, which I used to help bring me up to speed on basic virology while driving from Queensland to Tasmania to take up a new position as a postdoctoral researcher working on novel viruses of economic importance to the Australian salmon aquaculture industry. I'm writing again today because I heard in TWiV 425 that you still haven't received 17 emails since you announced that you had another book to give away. I do love free stuff, so consider this my entry into the competition. But I also had a virology question that ironically has sprung to mind while I've been listening to the TWiP podcast rather than while I've been listening to TWiV. I find the complex life cycles of many of the eukaryotic parasites fascinating. I think that the pseudophilidian tapeworms and trematodes in particular are amazing and both beautifully illustrate the interconnectedness of different organisms in ecological systems and the incredible power of evolution to find solutions to the problems involved in exploiting even the most apparently obscure niche. It boggles my mind to try and imagine how these parasites have evolved to be completely dependent on not just one, but sometimes two or three disparate host species for completion of their life cycles. While marveling at the incredible complexity of the life cycles of these worms with all their different stages, I started to wonder if there are any examples of viruses that have a requirement to infect more than one host species or cell type in order to complete their replication cycle. Of course, there are vector-borne viruses which need to infect an arthropod host in order to spread, but these viruses are still able to replicate in just one type of host. These viruses are still able to replicate in just one type of host cell. Are there any examples of viruses that replicate in a definitive host or cell type, but only if the virions have been somehow processed or modified in another intermediate host or cell type first? The closest example I could think of is the way HIV hitches a ride on dendritic cells in order to more effectively infect its definitive T cell host. But my understanding is that attachment to a dendritic cell isn't actually required for T cell infection and subsequent viral replication. I could imagine a scenario where virions produced in cell type A need to be modified by proteases in cell type B before they can reinfect another cell type A, or perhaps where cell types A and B need to be infected simultaneously in order to produce different components of the infectious viral particle. I can also imagine that if viruses with complex replication cycles like this did exist, they would be extremely recalcitrant to culture in the lab and hence would evade discovery in the first place. I would be very interested to hear your thoughts. Cheers, Scott. P.S. It's now the middle of summer in Tasmania, and the temperature here is a warm 27.5 C, wind from the northwest at 12 kilometers per hour, relative humidity 43%, dew point 14.1. And Scott is a research microbiologist in the Fish Health Department in Kings Meadows, Tasmania. Okay. So, do we know of any life cycles of viruses where you're obliged to go through one host before you can infect another, or one cell type before you can infect another?
I don't know of any.
I can't think of any right offhand.
I mean, this definitive and intermediate host thing is a feature of parasitic infections. But viruses, like you said, you typically have one host, and you can have multiple hosts, but you don't need to go from one to the other. The whole cycle is completed in the cell of one host. So, not that there aren't any out there, but we haven't found them.
Right.
Yeah. So, for example, in parasites, there's a mosquito cycle, and you have different forms maturing, and then those are passed on to another host where it does something even different. But for viruses, if you think of mosquito-borne viruses, they replicate in the mosquito, they replicate in the mammalian host, say, and the end result is the same thing, the same virus. So it's very different.
It's adaptive for protozoan parasites because their replication is not as simple as viral replication.
Be careful. Be careful who you insult.
Well, no, what I'm saying is a virus needs to get into a cell and replicate there. A protozoan can't just go into any old cell and take over and replicate there. It actually has to reproduce, and there are different immune systems that it would be interacting with if it goes through, say, an insect and a mollusk and a human. And because of those radically different environments, it has radically different adaptations to each of them that restrict the order in which it can go through those steps.
And remember, there are also sexual stages in many parasitic infections.
Yes.
And they're completed in one host and not the other.
It would reproduce sexually in one host and asexually in another. So there's just a kind of a bigger evolutionary space that it needs to explore in order to be able to adapt to whatever transmission cycle it's working out. Whereas viruses tend to have a more direct approach.
It's a good question, though.
It is a very good question, and I'd be fascinated by an example where there's an obligate intermediate host.
Yep. Well, we'll keep looking, but so far, no.
Listeners, please let us know.
Yeah. All right. Let's move on to some picks.
Okay.
Excuse me for the pause while I scroll down.
Yes. We don't have too many letters in the queue. Is this all of them?
There are a few left.
Okay.
Let's see. There are 1, 2, 3, 4, 5, 6 letters left. Yeah, and there will be more by next week.
Oh, sure. But
it's not bad. We've caught up. Yeah.
Previously.
Alan, what do you have?
I have actually a topical pick for today's show. This is I read this a few months ago, and it's been in my queue for a while. I said, "Oh, that would be the perfect thing to pick for this episode." It's a book called Deerland by Al Cambrone, and it is about deer, America's deer population, and our historical obsession with them, which goes back as long as there have been humans in the Americas. And it is if you're not immersed in this already, it is a surprisingly deep subject. So there's little unevenness to the book here and there, but it's really just an interesting exploration of what he refers to as the deer industrial complex. Which is the whole. There is a multi-billion-dollar industry built around various aspects of deer. People like to feed them in their backyards so they can see them. People like to hunt them and eat them. People raise them on farms so they can eat them. And this has been a thread through American history that has influenced our entire approach to wildlife management.
And then there are people who don't want them because they eat up their gardens and their shrubs.
Which is also extensively discussed in the book. Why do we have an overpopulation of deer? Well, it's because in many parts of the country we wiped out their only other natural predators, and now we are shirking our responsibility by not preying on them at the same rate. And just a really interesting analysis of the whole situation. It actually kind of made me want to take up hunting. It's that interesting.
Nice pun in the subtitle: America's Hunt for Ecological Balance.
Hunt for Ecological Balance.
That's probably the biggest roadkill I see here in the Northeast. Deer.
Yes.
All over the place.
Nationwide, they kill a lot of people and cost many, many millions of dollars in damage from just that.
And they're proliferating. I see more and more of them every year.
Ann Arbor's had
problem.
Ann Arbor's had a culling protocol with lots of people objecting to that, and
And people have tried to come up with other solutions. There are a couple of experimental birth control approaches that have not really worked out at all in the field, and it is a big and ongoing problem.
Yeah. Rich, what do you have?
I got two, so I'll be quick. One is one my wife found for me that was written up in the popular media called, it's a New York Times article called Extra Letters Added to Life's Genetic Code, and I also give the source, which is a PNAS paper. And apparently these guys, I didn't know about this, apparently these guys in the Venter Institute have been working for nearly 15 years on trying to introduce a novel base pair into DNA. And the motivation is, they say, expanding the genetic code. They're messing with, artificial life. And that's kind of the general area. It is a little obscure to me how you immediately translate this to expanding the information capacity of genetic code. But anyway, what this is some new papers on this, a new paper on this very topic, where they've actually created a novel base pair that they can stably incorporate into DNA. And these are wacky bases. They don't even have hydrogen bonding. They're maintained by stacking and hydrophobic interactions. But they do base pair with each other in order to, they're put in a non-essential region of a plasmid, okay? And in order to actually keep them there over generations, this is what this paper is about. They've had to put in transporters that will transport them in. The bases, I think, occur in, these are nucleotides that, or nucleosides that occur in some other organism. So they have incorporated transporters into bacteria that will transport them in. And they even stuck in a CRISPR system that is targeted so that if you mutate or delete these bases, it'll destroy the plasmid, which carries some sort of selective marker on it. So they've forced these cells to keep this base pair around. But when they do, it faithfully base pairs and will stick around for a long time. Where this is going to go, I don't know, but it was kind of interesting.
Hey, is this an orthogonal base pair?
Probably.
But it's being used in the same system, or is it? So it's not in regular DNA. It's in some separate piece of DNA in this organism.
Yeah, they're doing it. This is in bacteria.
Yeah.
And they're incorporating a single base pair into a plasmid.
Right? A single base pair.
Yeah.
Oh, I see. Not just, not multiple copies of it either.
No. And that, this is, after 15 years, we're still taking baby steps with this. Okay.
Hey, how long did it take to get that first triplet decoded?
That's right.
What was it? UUU or something like that?
I think so.
I think the whole point of this project is so that they can eventually produce a genome that's made out of something besides A, T, C, and G, and then they can make the announcement, it's life, but not as we know it.
Yeah, like
maybe. Would be interesting. I'm all for it. And you had a second.
I just ran across this morning, the Doomsday Clock.
Yes.
This is, I was unaware of this. This is a thing that was started in 1947 by the publishers of the Bulletin of the Atomic Scientists. Mm-hmm. And at the time when they originally published it, their concern was that because of the nuclear arms race, we might annihilate ourselves. And there's actually, there's somewhere in this link you can find out how close we have been to midnight over the years. This came up recently because they've moved. The closest we've ever been is 2 minutes to midnight in like 1957, when there was a lot of scare about the H-bomb. Okay. The furthest we've been is like 12 minutes to midnight, 1963, again in 1972.
There's 1991 with 17 minutes.
Oh, okay.
Yeah,
Pretty
when the Cold
safe.
War was over. Yeah.
Yeah.
It's been at 3 minutes for a while, and they just moved it up to 2.5 recently. They've now started to include in their predictions not just nuclear issues, but global warming and uncertainties about our political future as it impacts on these other factors.
So these are all predictions, right?
Oh, yeah. These are just a bunch of physicists getting together and saying, we're really nervous about what's going on here, and this is a way to express our nervousness.
Very interesting. I could definitely get into this. Wow, that's cool. Thank you, Rich. Kathy, what do you have?
I have something about noise cancellation, and it came up because I was at the dentist, and they were doing some drilling on the side of my head where my good ear is. So I whipped out an earplug that I always carry with me. And then the dentist and the assistant both admitted that they think that they may be losing some hearing because of the drilling. And I just thought, you should be wearing headphones or something. But what I wondered was, if I had noise cancellation headphones, would that actually cancel the sound waves, or would it only cancel the perception of the sound waves? And so I mentioned this to my brother, and he gave me these two links that were about unringing a bell and about noise cancellation, and some pictures of the physics of it and so forth. And the idea that this big bell in some town in Asia was waking up this child. And so then they show this experiment of using the noise cancellation and it then ringing the bell, and it doesn't wake up the child. So I thought it was really interesting. And even just an aside, putting the earplug in, of course, didn't help, because a fair amount of the drilling just travels through your bone.
The noise is coming from inside the head. Yes. These are cool videos. I like the explanation of the physics behind it, and then the one from GE where they're showing the actual experiment is very nicely shot as well.
I have two picks. Cool. One is a Nature article entitled "Rumors Swirl About Trump's Science Advisor Pick." Climate skeptic William Happer and ardent critic of academia David Galerntner have met with the president. Always the extremists have to be considered for all of Trump's committees. So these are two people who, have extreme views. They're part of academia, yet they don't believe in climate change. And the other one says, I have one quote here I pulled out. He says, "Science has never been less articulate, less interested in communicating its own excitement and wonder and fascination." And this is pure bullshit, because this person has no clue what's going on the internet with social media and how many scientists are engaged in communicating. Pure BS. I can't take this because it's just not true. It's fake. Oh, it's
an alternative fact.
An alternative fact. What did his press secretary say? Some facts are just not facts. Oh, right. This is so untrue. There's so many people excited about communicating, even old guys like us, even old folks like us. So this really bothers me. But, as my colleagues say, he will have what he wants. That's the way it goes. And then more recently, I'm sure everyone has heard about how all the federal websites, the EPA site and others, they've been taking down information. And this is an article in Scientific American. National Park Service won't be silenced. One of the first things they did was to take down data on the National Park Service, USDA website.
Well, and the GOP-controlled Congress also voted to devalue the national parks, which is presumably a prelude to opening them up to drilling or whatever they want to do.
Very nice. So,
it's a team effort going.
So the National Park Service, some of their employees started an alternative Twitter feed, aside from the real one, called Alt US Nat Park Service, and they are, they're not going to keep these data from being disseminated. We're going to
or just the tweets, and there's now twice as many people subscribed to the alt site as to the original site.
Well, on the tweets, they're going to be putting out the climate data, the CO2 index, and all of that stuff that they can't be doing on the regular Twitter feed. Yeah, I mean they can't. I guess they could put up an alternative website as well if they wanted to. But this is good. It's like follow up to you.
Yes.
Yeah, you found a whole bunch of other things.
Oh,
too.
yeah. So there's an alternative HHS site, alternative FAA, at alt_NASA, at rogue NASA. Alan pointed out at rogue_FAA, and there's an article in Forbes that talks about all of these rogue sites, and they have some interesting observations about the fact that many of these accounts are using copyrighted or trademarked logos for the official government organization, and so far contacting Twitter to ask them how do they police that. They haven't gotten answers from Twitter and so on. Just another way of a form of protest. Yeah.
Yes, but given that these are specifically political protest feeds, I think this would fall fairly clearly under fair use. I'm not an attorney, of course, but I think you could make the argument that since these are intended as political protests, they may need to alter the logo in some way, but it's certainly permissible to use the logo of what you're criticizing. The one I was most shocked to see was the FAA one, because most of these are purely science or largely science organizations, and scientists, of course, we aren't going to easily be censored. The FAA is a much more rule-bound type of organization, which is the way you want it to be. So it'll be interesting to see if that's for real.
I really doubt that most of the voters in this country really want to mine the national parks. They're so beautiful. I'm sure.
No, of course they
most
don't.
People.
And most of the voters in this country did not vote for the current president.
Yeah, but we have a stupid system in place that allows that, as you know. But the national parks are a treasure, and I hope it's above politics. I don't understand what they're doing. Unbelievable.
Yeah.
Embarrassing, actually, right?
Yes.
Yeah.
I have a couple of listener picks. One is from Stefan.
That's the one from above. That we read. I pasted it down here.
Thank you. Which may be more suited to TWiV than TWiV, but I feel the need to offer something with my first email. I just finished The Vital Question by Nick Lane, a biochemist at University College London, which is a really interesting discussion about the origins of life, particularly the circumstances which may have given rise to the last universal common ancestor, Luca, and the last eukaryotic common ancestor, Luca and Lika. I found the book fascinating, and I'm sure the Twix listeners would like it too. Sounds great. I'm getting it for sure. Except I'll get the Kindle edition, of course. We don't want any more paper here. I'm trying to
give them away.
Trying to get rid of my books. Not get rid, give them away to better hands. Kevin writes, Dear TWiV profs, I figure y'all might find this piece interesting. Thoughts on scientists being in the public sphere? I would normally say no, but the times they are a-changing. And this, of course, is an article about the impending march of scientists on Washington and other places that we've talked about. I don't know. It's a good question. Scientists Paul Ryan say that scientists have no right to speak out about policy or something like that? Is that what he said?
I'm sure he would say whatever was serving his purposes at that instant. But the scientists have always been in the public sphere.
Yeah.
This has been, this is something that scientists do and have advised policy for as long as we've had science, and this has led to conflicts when the facts are at odds with what the prevailing powers want to believe. And this goes back at least as far as Galileo. So, yeah, scientists should be in the public sphere and should take an active role in ensuring that we base our decisions on data.
Yep.
To the extent possible.
Interesting times for sure.
Yes.
And the last pick is from Dennis, who writes, "Hi docs, at onezoom.org there's an expandable fractal chart of the tree of life. On an iPad, for example, one can explore by panning and zooming. It takes a bit to get used to it, but the amount of info is tremendous. Who would have thought that fractals would become commonly used for big data display and visual search?" Maybe this could be posted as a pic. I'm just looking at this because I tried his original link and it was wrong, but onezoom.org seems to be right. Yeah, here we go. The best interactive tree of life ever. So here we have.
We've seen this before.
You think? Yes, you can click and zoom in more and more detail. Oh, this is wonderful. Look at this. Cool.
Maybe we haven't seen this one. Okay.
Oh, this is pretty neat. Like it. Thank you.
Sponges.
Neat.
You can look at sponges like might be in the Caribbean near Key West.
Very cool.
Yeah.
Thank you, Dennis. All right, if you want a copy, I have another book to give away. This is a good one. They're all good. And they're all new. I'm not giving away my old books. This is called Emerging Infections, just published by the American Society for Microbiology. It's edited by Michael Sheld, James Hughes, and Richard Whitley. And it has chapters on, about half of them are on viruses and half of them are on bacteria, for example, Ebola. Bunch of articles on that. MERS, Entros, D68, influenza, measles, chikungunya, Zika, West Nile. Then we have Chronobacter, Clostridium difficile, Bordetella pertussis, fungal infections. They even have chytridiomycosis, amphibian fungal disease. It's pretty cool. Brand new, just published. Very interesting stuff. If you would like this, today is the 27th of January. That's it. Be the 27th emailer. Now, everyone writes and says, I didn't think I had any chance. So nobody writes. So just write right away. And I'd like to get this over in the first few days and not have it go on for 2 weeks. So, right.
Yeah, it gets confusing if it goes on too long.
Yeah, then we have overlapping reading frames. Overlapping contests. Now I have TWiM and TWiV. Wow, can you imagine? All of them have overlapping contests, but I'll keep track of it. So the 27th emailer, twiv@microbe.tv, will get a copy of Emerging Infections. Just send an email with the subject line, Emerging Infections. And you know, some of you don't do it quite right, and I'm okay with that, but you should. Put it in the subject line like I tell you, please. Some people put something else in the subject line. It's funny. It's the way life is. All right, TWiV 426. You can find it at iTunes, microbe.tv/TWiV. Send your questions and comments to twiv@microbe.tv. Consider supporting us and our work. Go to microbe.tv/contribute. Different ways you can do that: Patreon, Amazon, PayPal, etc. We appreciate your help to let us travel around and be together more. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Rich Condit, formerly University of Florida in Gainesville, emeritus professor now in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time. I do want to point out in signing off that I haven't kept you up to date. The Vendée Globe, the first 6 sailors have finished. The first in record time. The second, my favorite, Alex Thompson and Hugo Boss, was a mere 16 hours behind the leader after going around the world single-handed. 4 others have finished. 11 out of the 30 boats have had to drop out, mostly because of mechanical stuff, and others are still pretty far behind, have thousands of miles to go. There you go.
Thousands of miles. Wow. It's not around the world in 80 days, is it?
It's close. 74 was the record.
Interesting. The song was right.
Yeah.
Alan Dove is at alandove.com. He's also on Twitter. Thank you, Alan.
Thanks. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the sponsor of this episode, Blue Apron. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.